Now I have the privilege of introducing our first speaker, Dr. Mario Masellis. Dr. Masellis is an associate professor of medicine at the University of Toronto and a clinician scientist at Sunnybrook Health Science Centers. He is, of course, also the co-leader of the ONDRI study. Welcome, Dr. Masellis.
So welcome, everyone. So I'll first start by introducing a little bit about dementia and what it is. There's a crisis brewing, the dementia crisis, and a tsunami is coming our way. And dementia is a leading cause of dependency and disability among seniors and can have a significant impact on social, mental, and physical well-being for patients and caregivers. I think that most people who are attending today has, in one way or another, knows someone who has dementia or family members who are providing care for those with dementia. It's touched 2 out of every 5 people. 1 in 10 Ontarians over the age of 65 have dementia. 1 in 5 Ontarians have cared for a person with dementia, and 1 in 3 seniors die having some form of dementia. So this is a real crisis that needs to be dealt with urgently. Dementia will affect an increasing proportion of the population, and this is really due to the growing age of our population and the aging of the baby boomers in particular. And this will ultimately result in an increased incidence of dementia. The strongest risk factor for dementia is not genetics, is not environmental factors, is not cardiovascular risk factors. In fact, the strongest is aging. So as people age, dementia is going to become more prevalent. And what this figure here is showing is that if you look at the current statistics, the majority of our population is kind of between the ages of about 40 and about 65. But in the next several years, by 2036, you're going to see a shift where the majority of the population is going to be in between the ages of 70 to 90. And as a result, dementia is going to be imposing a greater burden on our society. The risk for dementia doubles every 5 years after age 65 to about 25% by the age of 85. And about a quarter of Ontarians will be expected to will be greater than the age of 65 by 2038. This is going to become an increasing burden that needs to be addressed. What is dementia? So it's an umbrella term that is used to describe a progressively worsening syndrome. A syndrome is a group of signs and symptoms that someone suffers from that occur together and produce a clinical condition. So dementia is a progressively worsening syndrome characterized by problems with memory, decision-making, speaking, and behaviors that ultimately result in difficulty with day-to-day tasks and a loss of function. And the key here is that it's progressive. So dementia will generally always worsen. And there are many different causes of dementia. Neurodegenerative diseases are the main cause, but these can also co-occur with cerebrovascular disease or stroke. And in fact, over the age of 65, even though in textbooks, Alzheimer's disease is listed as the most common cause of dementia, in fact, over the age of 65, most commonly it's mixed diseases. So it's usually a combination of Alzheimer's disease combined with small vessel disease of the brain or Lewy body dementia that are the most prevalent forms of dementia. So as you can see here, based on this image, neurodegenerative diseases are the strongest risk factor for the development of dementia. However, what we see in life when someone comes into my clinic and I diagnose someone with Alzheimer's disease, we don't always get it right. And I'll be going into more details on this in the next slide. So the only way of definitively confirming if someone has Alzheimer's changes in the brain is currently on autopsy or pathology. After someone has passed away with one of these conditions, we're able to do an analysis on the person's brain and be able to confirm what the underlying changes in the brain are that are actually contributing to their clinical syndrome of dementia. So the concept here is that dementia is diagnosed in life by doctors and physicians, and we have a certain degree of ability to get the diagnosis right in life as to what the underlying causes are. However, we don't always get it right, and what we can't predict right now are those that have mixed pathologies or mixed causes in the brains. So what this slide is showing here are the neurodegenerative pathologies or proteinopathies which cause dementia, and these are the most common causes of dementia. And when I talk about proteinopathies, almost all dementias are caused by the accumulation of proteins in the brain, and proteins are the building blocks of our cells. And with the accumulation of these proteins in the brain in an abnormal way, because these proteins normally help the brain function properly, what happens is there's neurodegeneration or loss of brain cells or neurons. And so these are the most common pathologies. Here I show you the typical plaques, amyloid plaques, and tau neurofibrillary tangles, which cause Alzheimer's disease. So this is the pathology underlying Alzheimer's disease. Next to it, I show you alpha-synuclein inclusions in the form of what we call Lewy bodies in the brain. And this is the main cause of Parkinson's disease with mild cognitive impairment, Parkinson's disease with dementia, and also dementia with Lewy bodies. And this is the second most common neurodegenerative cause of dementia. In the slides below, we show the different causes of frontotemporal dementia, or FTLD, which is frontotemporal lobar degeneration. And there are two major causes, the tauopathies, and tau is a protein that goes awry in this disease, in these groups of diseases, and then the TDP43 proteinopathies, which not only can cause frontotemporal lobar degeneration or frontotemporal dementias, but it can also produce ALS as one of the most prevalent causes. So these are the major pathologies that we see as causes of dementia. And just to remind you, if you're over the age of 65, you're more than likely to have two or more of these pathologies in combination rather than one. We also must not forget cerebrovascular disease, or what we call cerebral small vessel disease, which can co-occur with all of these neurodegenerative pathologies and thus alter and change the progression rate of dementia and the way it presents. So dementia overall is extremely complex, and it's difficult to diagnose because of this complexity. And as I had alluded to, the clinical diagnosis does not always match what the pathology is. So in other words, if we have someone who is clinically diagnosed with Alzheimer's disease, they may have Alzheimer's pathology, but they may have other pathologies that we were not able to predict. Unless we follow patients over time, it may become more evident that there are other contributing diseases. The other thing that's very helpful in diagnosing these other contributing diseases are modalities such as brain imaging. So if we do an MRI scan, we can see hardening of the arteries in the brain in the form of small vessel disease and know that someone may have small vessel disease combined in. So we need to do research that helps us understand the relationships between the different stages and severities of the disease, because right now they're not well understood or associated in a linear fashion. So the ONDRI study wants to study these clinical syndromes, understand more about the underlying pathology that causes these clinical syndromes. And there's been a shift. We are starting to focus on biomarkers, and I'll go into a little bit more about this, and Dr. Munoz is going to go into a lot more detail about this. But biomarkers, the hope of using biomarkers is to be able to better refine our clinical diagnosis to be able to identify those that have pure forms of dementia, such as pure Alzheimer's disease, from mixed forms, such as Alzheimer's mixed with Lewy body disease or Alzheimer's mixed with small vessel disease. Ultimately, by understanding this complexity here, we want to understand how dementia functionally impacts patients and their families, as well as their free living behavior, such as sleep, exercise, social interactions. And this is another biomarker that Dr. McElroy is kindly going to be presenting on a little bit later on. So I alluded to biomarkers, and now I'm going to give you a definition of what biomarkers are. So biomarkers are needed to unravel the complexity of dementia, because if we just see someone in our office, we're not 100% certain whether or not they have Alzheimer's disease or Parkinson's disease, or mixed forms of the disease. So we need biomarkers, which are characteristics that can be used to objectively measure and evaluate an indicator of normal biological processes or pathogenic processes. And what I mean by that are the underlying changes I was alluding to in the brain. And ultimately, biomarkers may be used to monitor response to new therapeutic interventions that are being developed. So what else are biomarkers used for? So we want to be able to identify people who are at risk of developing disease, so disease susceptibility, or a trait that they may possess that puts them at a higher risk of developing a disease like Alzheimer's disease and developing dementia in general. We want to be able to use these biomarkers in the prodromal stages, so the stages before the full disease manifests, where, as someone's aging, they may have some very minor symptoms that may be just attributed to aging, but may also be attributed to pathological or abnormal aging. And so we want to be able to use biomarkers to determine if this is just kind of a change related with the normal aging process, or whether or not there may be an underlying pathology contributing to it. In someone who has full-blown symptoms of the disease, we want to be able to add biomarkers to the clinical diagnosis to state with more certainty whether they have Alzheimer's disease, whether they have mixed forms of dementia. We want to use this to help refine our differential diagnosis. We want to use biomarkers to track the rate of disease progression to identify people who will be more likely to decline rapidly versus people who have a more slower, indolent process. And then ultimately, when we have the holy grail available, that is disease-modifying therapeutics, we need to use biomarkers to track response. Ultimately, by adding biomarkers into the ONDRI program, what we're hoping to achieve here is personalized or precision medicine. So we want to be able to look at an individual and to be able to define what the underlying contributing factors are to their clinical neurodegenerative dementia syndrome, in order to be able to better guide and target therapies that are hopefully coming down the pipeline. So these are some examples of biomarkers that are already available in Alzheimer's disease. Unfortunately, they're not readily available in the entire healthcare system. But one biomarker is amyloid PET. And if you look at an individual who does not have Alzheimer's disease and compare it to an individual who does have Alzheimer's disease, and you use a special radioactive ligand that's injected into your veins and taken up by the brain, what we see here is that Alzheimer's disease have this kind of fluffy enhancement of their brain imaging, their PET imaging pattern, and that indicates that they have Alzheimer's pathology. If you look at structural MRI, so this is the sequence that we get in individuals who are participated in the ONDRI 1 program. What you can see here is that this is a control brain, and you can see there's very little shrinkage of the brain. But on this slide, you can see there's a lot more black, and the black is the space around the brain and in the center of the brain. And this represents atrophy or loss of brain cells or death of brain cells. And so when we see shrinkage of the brain, that's indicative of significant neurodegeneration, and different patterns of shrinkage are suggestive of different neurodegenerative diseases. Finally, there's new work being done with other PET imaging tracers. These are tracers that can highlight different proteins in the brain. And now we have tau PET available in the research setting that can indicate if someone has the tau conformation that's typical of Alzheimer's disease. We also have available biomarkers in the cerebrospinal fluid. So this is taking a sample of spinal fluid from someone's back, and we can send it to labs to determine if they have amyloid and tau pathology indicative of amyloid changes in the brain. And Dr. Munoz is going to be talking a little bit more about this. So there's an urgent need for disease-modifying therapies targeting these neurodegenerative pathologies, and we need biomarkers in order to be able to define what the underlying pathologies are in individuals who suffer from dementia. But as you probably are all aware, essentially all disease-modifying therapies have failed the gold standard of what we call the clinical trial, where we randomize a group of people to receive a drug, and we randomize a group of people to a placebo, which is essentially, a salt injection or a sugar injection or a sugar pill. And what we hope to show with these clinical trials is whether or not the disease group does better than the placebo group. Now, essentially, all clinical trials of Alzheimer's disease have failed, and I know that people have heard about the aducanumab story, which also has been a very controversial decision to approve in the US by the FDA, because most clinicians, when we look at the data, it doesn't seem to benefit patients. It may show some changes in biomarkers in the brain, but it doesn't seem to benefit the patients. Ultimately, what we want to be able to do is to figure out why these clinical trials have failed. And one reason why these clinical trials may have failed is because in life, we were not able to predict the specific pathologies that were causing the dementia syndrome. And as a result, even if we gave someone an amyloid drug, we didn't address their tau pathology, we didn't address their Lewy body pathology, or we didn't modify their cardiovascular risk factors, which caused, silent strokes in the brain. Ultimately, we want to use biomarkers to assist with defining outcome measures. We want to identify slow from fast progressors. Another reason why clinical trials have failed is that they've been given to people who are in the later stages of the disease. And once neurodegeneration, once there's been significant loss of brain cells in a person suffering from dementia or any neurodegenerative disease for that matter, it's probably too late for any of these drugs to work, even if they do appear to remove amyloid or to remove tau from the brain, for example. So we need to understand more about the basic disease mechanisms. We need to understand genetic risk. We need to understand and address other risk factors such as cardiovascular risk, so hypertension, diabetes, high cholesterol, smoking. These are all ways we can intervene to reduce the risk of developing dementia. So what do we hope to achieve in ONDRI? So the mission statement of ONDRI is that we're devoted to improving the diagnosis, the prognosis, the care, and outcomes for people living with neurodegenerative diseases, such as stroke and dementia. There are several different aims that we have. The first aim is to take advantage and analyze all of the data that we collected during the first phase of ONDRI, and I'll talk a little bit about that in the next slide. The second aim is to look at wearable devices that can be worn by patients at home to help track how people are doing with the disease, help to track their activity, their sleep, and to assess health and function in a person's home and community. And an extension of this is the Hands and Terra program, which will be discussed a little bit later. We want to not only work in isolation. In order for us to overcome all of the challenges and complexities of diagnosis, we need to collaborate. We need to bring our ONDRI data set into the space of other large dementia data sets, large neurodegenerative disease studies, in order to combine our efforts, to increase our sample sizes, to combine our data in order to achieve what we call higher statistical power, which allows us to better easily detect small signals in large samples, which allows us to be able to understand more about individual risk for disease, understand a bit about the basic science underlying this and how this applies to the health system, which relates to aim 4, being able to harness the power of data that's collected across Ontario at hospitals through pharmacy records, by analyzing health system records. And this allows us to track progression of disease. And so Laura and Dr. Bronskill will be discussing a little bit about this and available to talk about this later. So where were we in ONDRI? So I'll first talk about the foundational study. We studied 520 participants with a variety of neurodegenerative diseases, so cerebrovascular disease, frontotemporal dementia, Parkinson's disease, with and without cognitive impairment, Alzheimer's and mild cognitive impairment, and ALS. They were followed over 3 years, studying many different platforms, so a clinical platform, genetics, cognitive testing, gait and balance assessment, eye tracking, brain imaging, retinal imaging, and we've added a neuropathology and biomarker core, which is a really exciting add-on to this original diagnosis. And now this ONDRI data is not only available for us to study, but it's available for researchers around the world to study. And this is an exciting breakthrough, and we hope to have many more publications coming out from this foundational cohort. So right now, we are only seeing the tip of the iceberg, which is the clinical diagnosis of dementia. And what we want to do is use all of these powerful biomarkers that are in development and some that are proven to be able to resolve the complexity underlying dementia, to better understand these diseases at the microscopic level in living humans. And so this is why we want to, we're collecting autopsies in patients who consent to provide them so that we can understand the pathology that causes their dementia syndrome. And in life, we're adding the genetics and molecular platform. So I'll stop there.